Hello friends, today we will learn about Yersinia, Pasteurella and Francisella. So here we will learn about its morphology, culture, biochemical reactions, resistance, antigenic structure, pathogenesis, epidemiology, laboratory diagnosis, diagnosis of plaque in rats, treatment, prophylaxis, and then we'll study about Yersinia pseudotuberculosis and Yersinia enterocolitica. Under this, we'll study about Yersinia pseudotuberculosis and then we'll study about Yersinia enterocolitica. Next, we'll study about Pasteurella multicida. Multocida. Other then we'll study about other Pasteurella species and then we'll study about Francisella tularensis. So let's begin. There are these are gram-negative short bacilli showing bipolar staining with methylene blue. They are non-motile except Yersinia pseudotuberculosis and Yersinia enterocolitica. These bacteria are primary pathogens of rodents and men are accidentally infected. They were previously considered together under the genus Pasteurella. Based on their cultural, biochemical, and antigenic characters, this group has been divided into three genera: Yersinia, Pasteurella, and Francisella. The genus Yersinia contains three medically important species: Y. pestis, Y. pseudotuberculosis, and Y. enterocolitica. The name Yersinia is given after Alexander Yersin, who discovered the plague bacillus. The genus Yersinia now belongs to the tribe Yersiniae and the family Enterobacteriaceae. The genus Pasteurella is now restricted to several related bacteria causing hemorrhagic septicemia in different species of animals and occasionally producing human infections grouped under a common species called P. maltosida. The genus Francisella, consisting of the single species F. tularensis, is named after Francis for the contribution on tularemia caused by this bacterium. Now let's know about Yersinia pestis. Yersinia pestis is the causative agent of plaque, formerly known as Pasteurella pestis, was isolated independently and almost simultaneously by Yersin and Kitasato in 1894. Now let's know about the morphology. Y. pestis is a short ovoid gram-negative bacillus about 1.5 micrometer into 0.7 micrometer in size, with rounded ends and convex sides, occur singly, in short chains, or in small groups. When stained with methylene blue, it shows bipolar staining with two ends darkly or densely stained and central area clear. Now let's know about pleomorphism. Pleomorphism is very common here. It is characteristically enhanced in media containing 3% sodium chloride. In old cultures, involution forms are very common: cocoid, club-shaped, filamentous, and giant forms. The bacterial body is surrounded by a slime layer. It is non-motile, non-sporing, and non-acid fast. Now, let's know about the culture. The organism is aerobic and facultatively anaerobic. The optimum temperature for growth is 27 degrees centigrade, but the envelope develops best at 37 degrees centigrade. It grows on ordinary media. Now, let's know about the nutrient agar. Colonies are small, delicate, transparent after 24 to 48 hours of incubation. Now let's know about the blood agar. Colonies are non-hemolytic and dark brown due to the absorption of the hemine pigment. Other characters are same as that of the colonies or nutrient agar. Now let's know about the McConkey agar. Colorless colonies are formed. Now let's know about the broth. In nutrient broth, a flocculent growth occurs at the bottom and along the sides of the tube with little or no turbidity. A pellicle may form later. Now let's know about the ghee broth. When the organisms are grown in a flask with oil or ghee floated on the top, the characteristic growth occurs which hangs down from the under surface of the oil, resembling stalactites. That means stalactite growth. Now let's know about the biochemical reactions. Why pestis? Ferments glucose, mannitol and maltose with the production of acid but no gas. The lactose and sucrose are not fermented. It is catalase positive, indole negative, urease negative, methyl red positive, Voges, proscore and citrate negative. Gelatin is not liquefied on the basis of fermentation of glycerol and reduction of nitrate. Devignant has distinguished three physiological varieties of Y. pestis. This typing is of epidemiological significance because of the different geographical distribution of the types. Now let's know about the resistance. The plague bacillus is destroyed by heat at 55 degrees centigrade in 15 minutes. It is easily destroyed by sunlight, drying and chemical disinfectants. Phenol in 15 minutes. It can survive for several months in the soil of rodents' burrows. It remains viable for long periods in cold and moist environments. Antigen structure. Let's know about the antigen structure. Plaque bacilli are antigenetically homogeneous, and serotypes do not exist. The antigenic structure is complex and about 20 different antigens have been identified. These include a heat labile protein envelope antigen known as fraction 1, or we can say F1. It is best formed in cultures incubated at 37 degrees centigrade. It inhibits phagocytosis. This antigen is generally present only in virulent strain. It has therefore been considered to be a virulence determinant. It has an important role in stimulating protective immunity in mice and man. The V and W proteins have been considered to be virulence factors as they inhibit phagocytosis. These are also formed by virulent strains of Y. pestis. Next point is virulent strains produce a bacteriocin, that means pesticin 1, fibrinolysin and coagulase. Other antigens elaborated by Plaque include murine and guinea pig toxins. The endotoxin is similar to that of other enteric bacteria. Murine and guinea pig toxins are active in rodents and guinea pigs. Their role in natural disease of man is not known. These toxins are termed as plaque toxins. Virulence has also been associated with the ability to synthesize purines. Production of colored colonies on medium containing hemine also appears to be associated with virulence. It appears that Plaque possess an unidentified surface component which absorbs hemine and basic aromatic dyes in cultured media to form colored colonies. Now let's know about the pathogenesis. Y. pestis is a natural pathogen of rodents and causes zoonotic disease called plaque. Infection is transmitted from one animal to another by the bite of flea. Two natural cycles of plaque exist, the urban and the sylvatic plaque. Urban plaque involves man and rodents, that means rats, like living with him. In addition, cat, goat, sheep, dog, and camel are also susceptible. Wild or sylvatic plaque occurs in wild rodents, most important being mice, squirrels, and chipmunk. Plaque is a natural disease of rodents and is transmitted to man by the bite of infected rat flea. The incubation period is 2 to 6 days. In man, plaque occurs in three forms: bubonic, pneumonic, and septicemic. Let's know about bubonic plaque. As the plaque bacillus usually enters through the bite of infected rat flea on the legs, the inguinal lymph nodes are involved, hence the name bubonic. Bubo means groin. The inguinal lymph nodes become enlarged and suppurate. Patient develops fever, chills, and malaise. Pain may accompany that bubo. The infection may stop up to lymph nodes, but often the bacteria reaches the bloodstream and gets widely disseminated. If the bubonic plaque is not treated properly, it may progress to meningitis after 7 to 10 days of the onset of bubonic plaque. Now let's know about pneumonic plaque. It is highly infectious forms of plaque involving the lungs producing hemorrhagic pneumonia. It can be transmitted from man to man by droplet infection and is virtually always fatal. Primary pneumonic plaque is rare, but it may sometimes occur secondary to bubonic plaque. Pneumonic plaque may also occur in epidemic form. Patient develops fever and cough with expectoration. Now let's know about septicemic plaque. The presence of bacteria in blood denotes septicemic plaque. This may occur as primary infection, but it usually occurs as the terminal event of bubonic and pneumonic plaque. Massive involvement of blood vessels results in hemorrhages in the skin and mucosa. Due to this manifestation, the disease is given the name Black Death. Mortality rate in bubonic plaque is 50 to 75%. In pneumonic plaque, almost 100%, and with treatment, it is 5 to 30%. Pestis minor or benign plaque is also seen in some cases during an epidemic, and only clinical presentation may be slight temperature with one or two swollen glands. Now let's know about the epidemiology. Plaque is a zoonotic disease and may become epidemic in the rat population. The infection may spread to domestic rats and man by flea bites or contact. The commonest vector is Xenopsila chiopsis, but other fleas like X. astia, Ceratophilus may also transmit the infection. Plaque is the greatest killer known to mankind. In 14th century, plaque pandemic known as Black Death is believed to have killed about a quarter of all mankind. Several pandemics have occurred between 1500 and 1720. Last pandemic occurred in 1894, causing more than 10 million deaths by 1918. During 1958 to 77, plaque occurred in 29 countries and India remained free of plaque from 1967 to 1993. An outbreak of plaque was reported from India in 1994. A total of 876 cases of presumptive plaque were diagnosed on the basis of serological tests. These cases were reported from Maharashtra, Gujarat, Delhi, Karnataka, Uttar Pradesh, and Madhya Pradesh. Outbreak started in Bid district, Maharashtra for bubonic plague and Surat means Gujarat for pneumonic plague. Of the total 54 fatal cases, 52 were in Surat. 1900 in 2002, a short outbreak of plague was reported from Simla, claiming 4 lives. Plaque has several scattered natural foci in many countries. It survives in wild rodents, occasionally causing human infections. At least 4 such foci of plaque are known in India. These include the region near Kolar, Bid, Latur belt in Maharashtra, from where the Surat epidemic started, Rohru in Himachal Pradesh, from where the 2002 Simla outbreak took place, and a small pocket in Uttaranchal. Now let's know about the laboratory diagnosis. Laboratory diagnosis of plaque includes diagnosis of plaque in man as well as rodents. Early detection of infection in animals will help in prevention of plaque. Now let's know about the specimens. Pus or fluids aspirated from bubo in bubonic plaque. Next point is sputum and blood in pneumonic plaque, blood in septicemic plaque, splenic tissue on post-mortem, CSF in meningeal plaque. Now let's know about the direct microscopy. Sputum and aspirate from lymph nodes are stained with gram staining and methylene blue. Gram staining shows characteristic gram-negative cocobacilli, with methylene blue staining bacilli show typical bipolar staining. Microscopic examination of buffy coat smear of blood may show plaque bacilli in septicemic cases. Now let's know about the culture. Culture is made on blood agar medium and incubated at 27 degrees centigrade. Biochemical tests are performed to confirm the diagnosis. On ghee broth, there is characteristic stalactic growth. Now let's know about the animal inoculation. Guinea pigs Guinea pigs or white rats are injected subcutaneously with exudates from bubo or with 24 hours broth culture. The animals die within 2 to 5 days. Postmortem reveals marked local inflammatory reaction with necrosis and oedema. Regional lymph nodes are enlarged and congested. Organisms may be demonstrated in smears from local lesions, lymph nodes, spleen, and heart blood after staining with gram and methylene blue stains. Now let's know about antigen detection. F1 glycoprotein Ag may be detected in aspirated fluid from bubo or sputum by immunofluorescence and ELISA test. Now let's know about the serological test. Antibodies to F1 antigen may be detected by passive hemagglutination or complement fixation test. Antibodies appear towards the end of the first week in the illness. Serological tests are useful for identifying plaque foci, as the tests remain positive for several years after recovery from plaque. It has a limited diagnostic value as the titrates reach diagnostic levels only after 2 to 3 weeks following onset of disease. Now let's know about the PCR. PCR means polymerase chain reaction. It is a rapid and sensitive method for diagnosis of plaque in clinical specimens and flea. Now let's know about the diagnosis of plaque in rats. Before examining rats, immerse them in disinfectant to kill any flea. Necropsy reveals enlargement of lymph nodes with periglandular inflammation, and edema. Generally, cervical lymph nodes are involved due to the tendency of the rat flea to bite rat's neck region. Other features include pleural effusion, enlargement of spleen, and hemorrhage under the skin and in internal organs. Specimens like heart blood, lymph nodes, and spleen collected from the animals are utilized for smear examination, culture, and animal inoculation test. Now, let's know about the treatment. Tetracycline is the drug of choice in both bubonic and pneumonic plaque. Early treatment is essential because of high fatality of disease. Streptomycin is very effective. Chloramphenicol and kanamycin are also effective. Now let's know about the prophylaxis. General measures. General measures such as control of fleas and rodents are of great importance. Spray insecticides like DDT to inside the rodents, burrows, and houses to kill the fleas. After the fleas have been killed, kill the rats with rodenticides. Attempt should not be made to kill the rats unless spraying has first been done. Otherwise, the fleas will jump from the carcasses of rats to man. Stores, especially near railway stations, airports, and seaports must be rat free. Otherwise, infected rats with rat fleas may travel along the cargo to far off places. Now, let's study about specific measures. Two types of vaccines can be used to kill killed and leave attenuated. Let's know about killed vaccine. Let's know about the killed vaccines. Killed vaccine is widely used for active immunization. It is a whole bacterial culture antigen of plaque bacillus. It is prepared at Hopkins Institute, Mumbai. The plaque bacillus is grown in casein hydrolysate broth for 2 to 4 weeks at 32 degrees centigrade and killed by 0.05% formaldehyde and preserved with phenyl mercuric nitrate. The vaccine contains 2000 million organisms per ml. Now, let's know about the dose schedule. 0.05 ml of 0.05 ml is injected subcutaneously, followed by 1 ml after 7 to 14 days. Next point is booster dose is given every 6 months. Now, let's know about the immunity. Immunity appears 5 to 7 days after vaccination and lasting for 6 months. Vaccination may reduce the morbidity and mortality in bubonic plaque but not in pneumonic plaque. Vaccine should be given to health worker and to those planning to visit plaque infected areas. Now let's study about side effects. Fever, headache, lymphadenopathy, and erythema at the site of inoculation are some of the side effects. Vaccination is not very effective, therefore even vaccinated individuals should also be given chemoprophylaxis when exposed to plaque. Now let's study about live vaccine. Live vaccines are prepared from two virulent strains of Y. pestis, Otense tisvides strain from Java, and Girard's EV 76 strains from Malagasy. Since live vaccines are difficult to prepare and carry some risk of unacceptable reactions, killed vaccines are recommended for general use. However, mass vaccine is no more recommended. Now let's know about chemoprophylaxis. Close contact of patients with plaque should be given a course of tetracycline, that is 500 mg 6 hours for 1 week. Now let's know about Yersinia pseudotuberculosis and Yersinia enterocolitica. These two organisms resemble Y. pestis in that they are small gram-negative bacilli with bipolar staining and reservoirs of infection are rodents, wild and domestic animals. They differ from Y. pestis by their motility at 22 degrees centigrade, non-capsulated, urease positive, oxidase negative and insusceptible to Y. pestis bacteriophage. Now let's know about Yersinia pseudotuberculosis. It ferments rhamnose and mellibiose. It is antigenically heterogeneous on the basis of somatic and flagellar antigens. It is divided into 6 serological groups and 9 serotypes. Y pseudotuberculosis exhibits antigenic cross reaction with Y. pestis as well as with Salmonella. Pseudotuberculosis is a zoonosis. The infection occurs in animals by alimentary route, resulting in epizootic and enzootic forms of disease. A wide range of animals such as guinea pigs, rabbits, bats, etc. are susceptible. The liver, spleen and lungs of infected animals show multiple nodules resembling tuberculosis lesions and hence the name pseudotuberculosis. Man acquires infection through ingested materials contaminated with animal feces. Patients develop acute mesenteric lymphadenitis. Occasionally, it may result in a severe generalized disease. The term yersiniosis denotes infection with Yersinia other than Y. pestis. Laboratory diagnosis includes isolation of the organisms from an excised mesenteric lymph node or blood and demonstration of antibodies in patient serum during the acute illness. Now let's know about the Yersinia enterocolitica. It is a gram-negative Coccobacillus and resembles Y. pseudotuberculosis in being motile at 22 degrees centigrade, but differs from it in fermenting sucrose and cellobiose and decarboxylating ornithine. It does not ferment mellibiose or rhamnose. Many strains are indole and VP positive. It is aerobic and facultative anaerobe. Optimum temperature for growth is 22 degrees centigrade. On blood agar, smooth translucent non-hemolytic colonies measuring 2 to 3 mm in diameter appear after 48 hours incubation at 22 degrees centigrade. On McConkey agar, it forms pinpoint non-lactose fermenting colonies. Y. enterocolitica has a distinct antigenic structure and so far 60 O antigenic group and about 20 heat-labile flagellar antigens have been identified. Most of the human infections are due to serogroup 03, 08 and 09. Y. enterocolitica has been isolated from a wide range of domestic animals. Disease occurs via fecal-oral route by contaminated food and water. In man, it may cause gastroenteritis, mesenteric lymphadenitis, and septicemia. It affects children and adults of both sexes. The incubation period varies from 5 to 10 days. The early symptoms consist of fever, abdominal pain, and diarrhea. Diarrhea is probably due to an enterotoxin or invasion of mucosa by the bacteria. The laboratory diagnosis consists of isolation of organism and indirectly by demonstration of antibodies in the patient serum. The organisms can be isolated from feces, blood, or from mesenteric lymph nodes. Now let's know about Pasteurella multicida or P. septica. A group of related bacteria isolated from hemorrhagic septicemia in various animals and birds had in the past been named according to their species of origin. P. boviseptica, P. lepsaiatica, P. abhiseptica, etc. But now they are regarded as strains of a single species designated P. maltosida. Pasteur developed first attenuated vaccine from P. abhiseptica, hence the name Pasteurella. Maltosida is a non-motile gram-negative rod resembling Yersinia but differs from it in being oxidase positive, producing indole and falling to grow on McConaughey's agar. The bacillus is usually normal inhabitant of respiratory tract of a variety of animals such as dogs, rats, cats, cattle and sheep. It may sometimes occur as normal commensal in throat in humans. Human infection is rare but it may cause septic wound following animal bites, meningitis following head injury, respiratory tract infections or appendicitis. The bacillus is susceptible to tetracycline, streptomycin, and penicillin. Other Pasteurella species. Let's know about the other Pasteurella species. P. hemolytica, P. pneumotropica, P. urea, and other three important species of Pasteurella are the other three important species of Pasteurella. Their pathogenic role in man is not clear. Now, let's know about Francisella tularensis. Pasteurella tularensis. F. Pasteurella tularensis, or in short F. tularensis, is a causative agent of tularemia, a major zoonotic disease of rabbits and other rodents. The disease was originally described in Tulare County, California. The disease is transmitted by the ticks and several other arthropod vectors among the rodents. Human infection may occur by handling or eating infections meat or drinking contaminated water. It may also occur through tick bites. It is a capsulated non-motile gram-negative bacillus measuring 0.3 to 0.7 micrometer into 0.2 micrometer. It has fastidious growth requirements and special media such as Francis blood dextrose cysteine agar is used for its isolation. Minute transparent colonies appear after incubation for 3 to 5 days. In humans, tularemia may present as a local ulceration with lymphadenitis, a typhoid-like fever with glandular enlargement, or an influenza-like infection. The disease has been reported from North America, Europe, and Asia. The organism is highly infectious and laboratory infection has been quite common. Laboratory diagnosis may be made by examination, smear examination, culture, or by inoculation into guinea pigs. Pus from local lesions, blood during bacteremia, and sputum are used for diagnosis. Patient's serum may be tested for agglutinative antibodies. Antibodies a titer of 1:80 or higher is regarded as diagnostic. Since F. tularensis shares somatic antigens with both Br abortus and Br melitensis, false positive reactions may occur with Brucella agglutinins. Streptomycin is the drug of choice. An attenuated vaccine is available which can be administered to persons who are subject to high risk of infection. Finally finished. Any questions regarding this? You can ask me in the comment box and subscribe my channel to know more updates. Thank you.